So good afternoon to everyone. For me, it's a good pleasure to welcome today Kiko Solé to give a lecture in this new Showcasing Discoveries webinar series organized by the Josep Carreras Leukaemia Research Institute. So for those of you who do not know Kiko has studied biology in the Universitat Autònoma de Barcelona and received his PhD in biology in 1988. From 1988, he was working in the Hospital Creu Roja in Barcelona. Then in 97, sorry, he moved to the Hospital de Mar. And then from 2000 to 2012, he was working as a chief of the section in the Laboratory of Cytogenetics and Molecular Genetics Pathology Department in the Hospital del Mar. Actually, Kiko is the scientific director of the Institute de Recerca contra la Leukaemia Josep Carreras in the campus ICO GITP. He is also an associate professor in the Universitat Pompeu Fabra and is a professor collaborator at the University of Barcelona and in the Universitat Autònoma de Barcelona. So Kiko has an extensive experience in the application of conventional cytogenetics and techniques and molecular techniques in the study of hematological disease. And his talk today will deal with the new discovery in the field of MDS. So before Kiko, before you start your presentation, I would just like to remind to the audience that you can ask your question using the button in the application. Then Kiko, so we are eager to know about your last discovery in the field of MDS. Thank you.
Thank you, Gael, for this nice and friendly introduction. Here are the topics that I will develop during my talk. First, who are we? Here I want to introduce my group. It's not a big group. It's a group that I am very proud because we are very active and productive. In green, the person that are PhDs, Mar, Mayo, and Laura are my left and right hands, and persons very involved in my team. Pami is the last PhD in our group that recently defended the doctoral thesis. Francisco is the bioinformatics and will defend the doctoral thesis as soon as possible. And Anna is the technician that without her, our research will be more difficult. And also I want to comment that Vera Dema arrived with me in 2012 from Hospital del Mar and was working with me until 2014, that she moved into Cleveland Clinic and now recently moved to MD Anderson that is working with Guillermo Garcia Manero. And also you have that without ECO, Service Hematology Department, our research is not possible. I have to stress the contribution of Lourdes Zamora, Blanca Chicoy, Isabel Granada, Javier Grau, Silvia Marceta, Marta Cabezón. Without them, our research is more easy. And also I want to remark, although my group looks big, it's not as big as I would like, because Laura is also in charge of single cell unit, a very promising unit, because I know that you have a very important presence in future. She will be very busy in this field. And Marmayo is the person in charge of microarrays unit, a unit that is very productive and very active. Regarding ongoing projects, I will explain in short vignettes what we have done during the last years and what we are doing in our department, in our group. And the aim of this presentation is that at the end of this presentation, that you could have a real image of the research that is done in our group. First, I want to summarize a few words about MDS, myelodysplastic syndromes. MDS in the past were called pre-leukemia because they have a high risk to transform into AML. It's a rare disease, like most of the hematologic cancers. Is an age-related disease. If finally we live more than 150 years, we will have a high chance to be diagnosed of MDS. The survival, it's bad news, is the same during the last years. The only curative treatment at this moment is bone marrow transplantation, but you cannot transplant people older than 65 years old, and most MDS are older than this age. At this moment, Caritab is a strong factor for prognosis, and here we have the EPSS arm that we were very active contributing. And we have to remember that 50% of cases present cytogenetic aberrations by karyotyping, whereas by sequencing 90%. And now we are also working in the International Working Group of MDS in the definition of a new scoring system based on molecular studies, APSS-M. I will try to develop my talk explaining the sequence or the multiclonal evolution disease. We have CHIP. CHIP is not a disease, perhaps it's a status. Its CHIPs are those, not patients, are those individuals that have the same mutations, same mutated genes that have patients of MDS. But if you have CHIP, you have a risk to develop, highest risk to develop leukemia. Then we have the MDS, and we studied in our group the low risk and high risk. And finally, the evolution of MDS could be into AML, the transformation to real leukemia, or to therapy-related myeloid neoplasms. Therapy-related myeloid neoplasms are those patients that have previously diagnosed of a previous cancer, solid cancer or metallurgic cancer, that are treated with chemo or radiotherapy that have a high risk to develop a secondary leukemia. In the first part of my talk, you will see that my research was focused in MDS of low risk and also on high risk. Our group is more dedicated to translational research. And in the last part of my talk, you will see that now we try to do investigation of all the spectrum of diseases related to this MDS. And I have to tell you, or to share with you, that I love working in this disease, but I think that the name is not very beautiful. I do not like very much the name. Why? Because when you have to explain to the citizens that you work in MDS, people say, what? What's that? It's a disease, it's a cancer, it's MDS, Myelodysplastic Syndrome, have a terrible name that is not very friendly to the population. First, I will start with explaining some projects related with an entity, MDS with deletion of chromosome 5, of the long arm of chromosome 5. 5q- is the most recurrent cytogenetic abnormality in MDS, followed by monosomy 7, 7q- or trisomy 8. If 5q- is present as a single aberration, confers a very good prognosis, and this is the low-risk MDS. And if it is associated with other alterations, could confer a poor prognosis, and this could be related with high-risk MDS. More than 10 years ago, lenalidomide has been approved for the treatment of patients with low-risk MDS with single interstitial deletion of the long arm of chromosome 5. And it was demonstrated in previous studies that low-risk MDS with 5q- treated with lenalidomide, 75 of them responded. And low-risk MDS without 5q-, just 30% of them were responders. So lenalidomide is a treatment very, very indicated for patients with low risk, 5q- as a single aberration, because most of them will respond. This response is translated with less transfusion requirements and with the less cells with 5q- deletion. And I want to comment first this paper. It was when I was working at the end of Hospital del Mar. And in this paper that was written by Mar Mayo, we collected the largest series of patients with 5q- at MDS, and we demonstrated something really simple. Patients with 5q- as a single aberration presented the same prognosis, survival, as patients with 5q- and one additional aberration. Whereas patients with complex karyotype, that those with 5q- and two or more cytogenetic aberrations, those patients were related with a very poor prognosis. This very simple permits or give to the WHO classification of 2017 change the definition, and they accepted MDS with isolated 5q-, those patients that presented 5q- as a single aberration or with one additional aberration except monosomy 7 or 7q-. So thanks to our contribution, we modified this entity, the definition of this entity. That is, at this moment, the only MDS entity associated with a cytogenetic aberration. Then we followed the study patient with 5q- treated by lenalidomide. Thanks funding that we received from Celgene, we studied 52 patients with MDS and 5q- treated by lenalidomide using conventional cytogenetic SNP-based genomic sequencing. At this moment, just Sanger, because it was not the era or was not, we did not have experience in next generation sequencing. We studied TET2, that was the most frequent gene in MDS, and TP53 because previous papers suggested the poor prognosis of patients with this gene mutated. And what we demonstrated is that the number of cytogenetic aberrations and the platelet count were the parameters that strongly explain the response or not response to lenalidomide in this group of patients. This is the good news. These two parameters are parameters easy to have in a routine lab of diagnosis of MDS patients. Do not require specific techniques. Then we focus also on studying the 5Q patients. We receive more money from Celgene company, and we receive a very big grant from Instituto Carlos III. And the objective of both projects, we asked the money at the same moment, and from both projects we have acceptation. We wanted to do exome sequencing of patients with 5q- treated by lenalidomide. We wanted to know if some genes could explain response or not response to this treatment. Thanks to these first studies, we published this very good paper in Cancer Cell, in which the second author was Vera Adema with Rebecca Schneider, and we detected a new gene, and was the first new gene located in the 5q region that was detected mutated in patients with MDS and 5q-. And I have to share our experience with these findings. We did sequencing of 3 patients. Among these 3 patients, in 2 of them, this gene was mutated. I would say, wow, you have the gene that explains 5q- cases. But the bad news is when we studied more than 40 additional patients with 5q-, this gene was not mutating in the other largest series of patients with 5q-. So the gene explains some cases, in some cases with 5q-, the pathogenesis of this syndrome, but not the pathogenesis of all cases with 5q-. Still, we need more research to know what are the other genes that could contribute to this syndrome. And then from this time until now, with a collaboration with International Working Group, in which Jaroslav Maciejewski from Cleveland Clinic, that is the boss, was the boss of Vera Adema, we follow to acquire information, and we have genomic data, RNA-seq, and clinical data of 388 Delphi patients, and we compare the genomic findings of this group of patients with 844 myeloid patients without 5q-. And this is the paper that was recently submitted. For this reason, I could not present results. Then we follow the research in patients with 5q-, because the Celgene company was really interested to know why patients respond and why other patients do not respond, and we receive money to study patients with 5q- and without 5q- all treated by lenalidomide. We studied those patients using a panel of genes that we designed it in the Spanish working group of MDS. And finally, we published this paper in British Journal of Hematology, and we found the following conclusion. DNA-mediated mutations were very frequent in patients with 5q- and predicted a good response to lenalidomide, whereas mutations of TP53 were observed on both groups of patients with 5q- and without 5q-, and in both groups of patients, the mutation confers a very poor response to lenalidomide. An interesting finding is that mutations of FF3B1 and TET2 were mainly detected in the group without 5q-, and these mutations confers a refractoriness to lenalidomide. These are some preliminary findings that should be confirmed in larger series. This is the next project in which we were involved. We finished the first fish grant, and this is another fish grant, and we wanted to analyze 5q- patients treated by lenalidomide using single cell. It was the moment that started to have single cell available in our research centers. And we wanted to analyze patients with 5q-, some responded and some not responded, and try to know the clonal architecture of these patients. The preliminary results were presented in the Spanish Hematologic Meeting in 2017, and the paper is in preparation, and we plan to submit the paper in the next 2 weeks, I hope. And here you summarize a little bit this project. From all these 4 patients, is 4 women in this case, we extracted peripheral blood and bone marrow. From peripheral blood, we try to have the T cells, and the T cells will be used as germinal controls DNA. And from the bone marrow, we could have the bone marrow, we will stack the DNA, and with this germinal DNA compared to the tumoral DNA, we perform exome sequencing and SNP arrays. With exome sequencing, we detected the genes that were mutated in those cases, and with SNP arrays, we detected copy number alterations. Then from the bone marrow, and with the sorter, we selected a population of tumoral cells, and these tumoral cells, knowing the genes that were mutated at the copy number alterations, we use it to perform single cell analysis. In this time, we use the Fluidigm platform that was the best platform at this moment. We know that at this moment you have better platforms and that permits to analyze more number of cells. And here I want to summarize one case of the 4 that we have analyzed, an interesting case. In this case, the case presented just 3 genes mutated and 2 copy number alteration. One was a 5' that was previously detected by karyotyping, and the other was a deletion of the short-term chromosome X that was just detected by microarray system. Okay, this patient entered in the SyntaRep clinical trial. SyntaRep clinical trial is a clinical trial in which patients, normally you could treat a patient with lenalidomide if patient have 5' and requires transfusions. In this SyntaRep clinical trial, you could treat patients with lenalidomide with 5' as a single aberration, but that do not need transfusion requirement. And here is a summary of this follow-up of this case. We have, here, we have a diagnosis of previous to treat the patient that have a clone with 5' cell as a single aberration, and then you have normal cells. And you doing the single cell study, we detect that the clone with 5' was not the ancestral clone. It's an interesting finding. That was a subclone. And in our experience with the paper that we have submitted to publish with Vera de Mar, we demonstrate that the 5' could be ancestral or subclonal. And in patients that is ancestral, the prognosis is better than in patients that is subclonal. But here is just one case. But this is what we know from our study of more than 300 cases. And in this case, that diagnosis have this big clone, another subclone with 5' another gene mutated, and a small clone that do not have the 5' cells. In those cases, the diagnosis could analyze 177 cells. With the new platform that we have now in the institution, you could analyze thousands of cells. So the conclusions of single cells will be more robust and more informative. This patient was followed by their treatment, that until now we don't know what treatment received the patient because was in a clinical trial. And after 14 months, the patient had that all the 20 metaphases analyzed were normal, and the FISH study analyzing 200 cells were normal. But it's interesting to remark that by single cell, we also still detect 5% of cells with 5' deletion. So single cell or sequencing is more sensitive than the conventional techniques. And then in the second follow-up, all the cells by karyotyping and FISH by FISH we don't know were normal, and by single cell, all the cells were normal. So we can see that we can suggest that this patient should be treated with lenalidomide because placebo will be not enough to make the disappearance of the clone with the cytogenetic alteration. This patient had a complete cytogenetic response, and we suspect we will see soon that the patient have received lenalidomide. Okay. Then I will explain some projects related with MDS, not just with MDS and 5' and receive funding from the Stiftung Josep Carreras Foundation. It's a project in collaboration with the German group from Göttingen, with Detlef Haas being the leader. It was a very beautiful project in which we wanted to analyze sequencing of the same patient using peripheral blood and bone marrow. sometimes it's difficult to obtain bone marrow, and we wanted to know if using peripheral blood, we could have the same results that we observed in bone marrow. From the same patient, we have the sample bone marrow cells, we have peripheral blood mononuclear cells, and from peripheral blood, with the sorted, we had 34 cells. Using this technique and a large number of patients, 48, we could demonstrate what the results were very comparative between peripheral blood and bone marrow, but the results were very similar if you use peripheral blood using CD44 cells. So in cases in which you could not have bone marrow, you could use peripheral blood to do sequencing, but in those cases, we recommend first to do a separation of CD44 cells. This is another project that is an ongoing project that will finish soon. Is in a proposal that was done International Working Group of MDS. 50% of patients with MDS present a normal karyotype, and some of these patients with normal karyotype could be studied by SMP arrays. And we collected the largest series of patients with normal karyotyping and studied by SMP arrays, 163. And by SMP arrays, we detected that 50% of those patients presented aberrations detected by SMP arrays, and some preliminary results suggest that the detection of LOH, copy number neutral, had prognosis impact, poor prognosis impact, as well the large copy number alterations. Small aberrations do not have a real good worse impact. And the idea is to submit this paper after some statistical analysis at the end of this year. And this is another project in which our team is involved. It is international working group in which we want to define the MPS scoring system based in molecular studies. The coordinator is Benjamin Eber and Eli Papanuil, and the funding comes from the MDS Foundation. And we are all members that are working in the MDS working group of MDS. And the results, I hope that we analyze will be presented provisionally in the ASH meeting of this year, that you know that will be virtual, not real. And here is some slides about this project that I received from Elsa Bernat and Eli Papanuil, that are the hard workers of this project. 13 countries are involved in this project. More than 25 centers are involved in this project. More than 4,000 cases are studied by sequencing. These are different working groups, and I am with Dr. Fras, the person in charge of the cytogenetics working group of this project. And some preliminary results. So the copy number alterations neutral in 11%. With cytogenetics, 44% cases presented aberration. With mutations, 91%. So a large number of aberrations could be detected just by sequencing. If we combine all the techniques, 95% of patients present genetic alterations in MDS. And here also the important findings that the number of genes mutated is related with the prognosis of the disease. More mutations confirms a poorest prognosis for the patients. And another interesting finding is SF3B1 cases as a single alteration present a very good prognosis. This is something logical because most of these patients are diagnosed with sideroblastic anemia, and sideroblastic anemia is a MDS disease that is related with a very good prognosis. And with this preliminary data, Elsa Bernat and Eli Papanuil analyzed that the status of TP53 was important to define the prognosis of these patients. And with this preliminary data, we published recently this paper in Nature Medicine. And the conclusions are very simple, but very interesting, because were the first conclusions that were demonstrated in a cancer that I am sure that now will be studied in different cancers, solid and non-solid. And here are the interesting findings. If the patient have one mutation of TP53, mutation could be mutation or deletion of the gene. If the patient have one allele affected or no mutations or not affected of this allele, the prognosis is the same, it's very good prognosis. Whereas when the patients present the two alleles affected or mutated, then the prognosis is very poor. It sounds logical because it's a tumor suppressor gene and it's important to have both copies mutated or affected. But this simple finding, we were the first that demonstrate this in a cancer, and I am sure that this soon will appear a lot of papers that will try to to validate or confirm these findings in other solid tumors and other leukemias. And here is another ongoing project that we receive a very huge funding from Celgene company. It's a very beautiful project that I like a lot. I will try to summarize here, in which we want to have sequential samples from patients with low risk. We will follow patients with low risk, and every 6 months we will try to find a sample of peripheral blood. Why peripheral blood? Because we know that using peripheral blood, we could have the same information of genomics than using bone marrow, and it's a less invasive tissue. And we will have patients of low risk. We will follow up during 2 or 3 years. Among these patients of low risk, some of them, we think that perhaps 10% of them will have a progression to a high risk or to acute leukemia, and the others will not evolve to acute leukemia. You want to compare those patients, the genomics of those patients that progress, compared to the genomics of those patients who do not progress. And we want to know if the sequential study of the genomic, if we perform sequencing in sequential studies, if this information will help to know which patients will transform and which patients will not transform. This is the hypothesis that we want to work. At this moment, we are collecting data and collecting samples. And here is another recent project that we received. This is an European project, TransCan. TransCan means Translational Cancer Research Project. The PI of this big project is Matteo Della Porta in Italy, and the collaborators is one from Germany, Uwe Platzbecker, one from France, Pierre Fenot, and me from IGC. We received the funding part from Instituto de Salud Carreras III, part from Asociación Española contra el Cáncer. And with what I can share with you at this moment, we have the information of 5,000 healthy individuals older than 16 years, also studied by targeted sequencing. We have more than 2,500 patients with MDS also studied by NGS. And what we will do in our center, in our group of research, we will perform single cell studies of patients with low risk that transforms to high risk. We have samples of patients with low risk and the same patient that transformed to low risk. We have those kinds of samples, so we will perform single cell studies in these patients. And also another interesting result that we will have, we have samples for patients that have clonal hematopoiesis or CHIP, and those patients that transforms to low risk MDS. It will be very interesting to know what happens in a patient that have low risk and transform to low risk, and what happens in a patient that, not a patient, an individual have clonal hematopoiesis that later develops MDS. With that, we will know what happens, why some patients have clonal hematopoiesis and why of them transform to MDS and why the others do not respond. And I hope that next year we could present results about these findings. Here are 2 papers that were submitted thanks to the collaboration of this European working group. This is the first one that was submitted to JCO, and yesterday we received a mail from JCO. You have this minor change. The change are really minor, so the paper will be accepted. And the other is a very interesting project in relation more with the clonal hematopoiesis. It's in a journal with a very good impact factor, and we are waiting for the news of the reviewers. But I think that this project, we will have a very interesting results. Here, MDS, and now we move to another MDS that is more rare than MDS. It's MDS MPN. It's a very infrequent disease, this is not very well known, and received a FIS project from Instituto Salud Carlos III in 2017. And in this group of patients, we try to have a larger series of patients, 50. We have 50 patients because every month we send an email to our colleagues in Spain saying, please, we need patients with this disease because it's very, very rare. And we do not want to lose any patient from Spain with this disease. And among these patients, we want to perform SNP arrays, targeted sequencing, whole exome sequencing, whole genome sequencing, RNA-seq from these patients. Now we are at this moment collected samples. We did some SNP arrays studies. We did some targeted sequencing. We did some exome sequencing. And soon we will start with RNA-seq studies. And the idea is at the end of this year, maximum next year, we will finish to have all the results of this project, because we have to finish this year. but we sent an email to Carlos Terteo saying we need to finish next year because by the COVID, we have some delay on achieving the results. And now I want to change the disease. This is the disease therapy-related myeloid neoplasm. It's the most aggressive part of this group of patients. Therapy-related myeloid disease, just to remember you, are those patients that had diagnosis as a leukemia, as a cancer, then are treated with chemo or radiotherapy, then are cured, and after a period of time that the patients were normal, they could evolve or transform to a secondary leukemia, or now we call therapy-related myeloid neoplasm. And inside the International Working Group, I am very involved in this, in defining how is this disease at the genetic point of view. And here we have the first paper that recently published. The good news of this paper was submitted one day and was accepted the day after. Was really fast. I don't know. I am really surprised, but I think I suppose that the editor liked very much this paper. And just to comment, what is the most important of this paper is that the WHO classification include therapy in MDS, therapy MPN, or therapy AML in the same entity of patients. All are classified as therapy-related myeloid neoplasm. And our findings in this paper suggest that we have to separate this entity in the smaller entities. One entity of therapy-related MDS, another entity of therapy-related MPN, another disease of therapy-related AML. This subclassification will be more useful for clinical treatment of the patient to do the better follow-up of those patients. And now with that left has, we are involved in another 2 papers or 2 projects using this database of therapy-related myeloid neoplasm. We want to define what are the cytogenetic alterations in relation with the previous cancer, hematologic or solid, and different types of hematologic, and in relation with the treatment previously received by the patient. Now we want to define if the karyotype could be correlated with the previous cancer and the previous treatment. There is a paper that is written by Meritxell Nomdedeu that in the past was working here in Nico Germans Tracy Pujol, and now is writing the paper. This summer did this. He was at home writing the paper, and soon we will do the first reviews, and I hope that soon will be published. And after this paper, we are planning to develop a new scoring system for this group of patients. Provisionally, I can share with you that IPSR works very well in this group of patients, but the cytogenetic stratification needs some kind of changes of genetic alteration, because some alterations works very well, but some others not. Most of them works very well for this group of patients. And then this is another project that I like. It's not a project for me, it's a project that I like, but it's a project in which it's really difficult to obtain money, because the project is not a short project. For me, it's a very long project that started in 2018, and I hope that will be finished a lot of years ago. And it's an interesting project that we wanted to analyze patients with the previous diagnosis of breast cancer, and also patients with the previous diagnosis of ovarian cancer that were cured. Among these patients that were obtained thanks to eco collaboration from the oncology department here, Mireia Marjalí and Marga Romeu from the oncology department. From this patient, every year we try to receive one sample from peripheral blood of the group of patients that were cured from their previous cancer. And after a long follow-up, we hope that most of these patients do not evolve into therapy-related myeloid neoplasm, but few of them, is the pity of what we don't like, but few of them could develop a therapy-related neoplasm. And our plan will be to demonstrate or try to know if we could have serial samples of peripheral blood analyzed by sequencing, we could help to know which patients have a high risk to develop therapy-related myeloid neoplasm and which patients will have not a high risk to develop this this poor prognosis leukemia. I have to share with you that CHIP or clonal hematopoiesis is observed also in myeloid diseases without disease, that adult patients diagnosed of cancer, solid or not solid, have a risk of developing therapy-related myeloneoplasm. All patients that receive chemoradiotherapy have a risk to develop therapy-related myeloneoplasm. And it's not interesting finding is a patient that have cancer before being treated. If you detect in those patients CHIP, clonal hematopoiesis without disease, those patients have a higher risk to develop a therapy-related myeloid neoplasm. And this is a pilot study that using patients with breast cancer for the reason you call mama sec in Spanish, or nobody sec for an ovarian cancer. And it could be interesting to know perhaps patients, all patients, my idea will be that all patients diagnosed of cancer, previous to treat the patient, it will be important to know if the patient have CHIP. If the patient have CHIP, then the patient have to require a less aggressive treatment. And if the patient do not have CHIP, then the patient could receive a more aggressive treatment. This is the idea to do this sequential study that with a long follow-up. We received the funding from the Rotary Badalona in 2018, and now we are collecting samples that what I want to share with you, these samples are in the biobanking or in our institution, but could be used for this project, or if you have any ideas related with this project, we are open to propose a collaboration with all of you. This is the summary or a diagram of this project. Our idea is at diagnosis, perform whole exome sequencing previous to treat the patient and targeted sequencing of all the patients with breast cancer or ovarian cancer, then treat the patient, perform another additional exome sequencing after being treated the patient, and then obtain sequential samples of peripheral blood, one sample per year, this will be the best, and to perform, and after a follow-up, a long follow-up, compare those patients that evolve into therapy-related and those patients that do not evolve into therapy-related, and compare the genomic of this patient. And we hope that using NGS in sequential samples from peripheral blood of patients treated, we could define which patients will evolve or not to therapy-related myeloid neoplasm. And here is the last project that we received the good news last month. This is our last phase project about the genetic characterization of therapy-related myeloid neoplasm. But this is part of an coordinated project that is around the MDS disease. In this project, the team is Isabel Granada from the Genetics Lab, Ico Germán Pujol, Mar, Laura, Silvia, Javier Grau, Pamela Hacha, Silvia Mar, repeated Silvia Marcet, Marta Cabezón, Francisco and Iris Teruel is the oncologist of this project. I am the PI. It's a funding that we received for our center, but we have done this project in collaboration with other teams from the Spanish working group of MDS, and we call this project Umbrella Project. The coordinator is Maria Díaz del Campero. Maria actually is the president of the Spanish working group of MDS. Felipe Prosper in Pamplona is also involved, Dave McCarthy in Vallebron, and we are here involved in this very nice project. We like this project. The idea is in a large number of MDS patients perform NGS studies to have clinical characterization, to define better the prognosis and to know the evolution of this disease. And each of the working groups of each city will do different things. Salamanca and Pamplona will do more basic research in MDS patients, and Vallebron will work more in the clonal hematopoiesis, and we will work in therapy-related myeloid neoplasm. But the idea is to correlate the findings of the clonal hematopoiesis studies on Vallebron with the findings that we will have with therapy-related myeloid neoplasm. And the idea is all different groups very strong in this field, in MDS field in Spain, work together to know the better characterization of this patient. We have here also the support of the Instituto Salud Carlos III, but behind also we have a large series of patients with MDS that are inside the Spanish working group of MDS. That the Spanish working group of MDS, I can tell to you, all of you, that at this moment we have 14,000 MDS patients registered in a database. We are the country with the highest database of patients with MDS. And now one of my aims inside this working group is try to have samples from all the cases that are now being diagnosed of MDS. So we have a good project working together in the Spanish working group of MDS. And here, just to summarize a little bit our part of the project of this FIS grant, we will perform whole genome sequencing, whole exome sequencing of 50 patients, 25 of them with complex karyotype and 25 of them with normal karyotype. You have to know that 90% of patients with this disease have a complex karyotype and just 10% will have a normal karyotype. So it will be more difficult to have 25 with normal karyotype. But if we collect patients from all Spain, we will reach this number, I hope, easily. These patients will be studied by a whole exome sequencing. And then among these patients, we expect that in 10 of them, we could have peripheral blood at the moment that the patient was diagnosed of the previous cancer. So if they have at least 10 patients, 10 samples of peripheral blood at the original diagnosis of tumoral cancer or hematologic cancer, then we will study these cases by targeted deep sequence, and we hope, we expect by the published information that at least 5 of them will have clonal hematopoiesis. And then our idea is to use single cell studies that we will have in our institution to know the intratumoral heterogeneity of this group of patients. We want to analyze those with normal karyotype, and we will want to compare with those with a complex karyotype. I am sure the clonal architecture will be totally different because the architecture will be different, also the clinical implications of having normal karyotype or having a complex karyotype is really very, very different. And now I'm in the last part of my talk, just a summarize, really summary of the publications of our group. I arrived here in October in 2012 with my 2 fellows, Mar Mayo and Vera de Mar. And from 2012 until now, we published 99 papers, and I am the last author or corresponding author in 38 of them. And here below you have the publication of each of my fellows, Mar Mayo, Laura, Pamela Hacha. Pamela Hacha now will start to publish more papers because she's preparing new papers. And Francisco Fuste is the bioinformatics that also have a very good curriculum. It's more than 10 papers in publications. And here is a summary of the last year. I think for my group is a very good year because until now we published 15 papers, 15 papers. In here is the impact factors the journal. I have 2 bloods, 3 leukemias, and journals of very important factor in the field of hematology and in cancer. And we also have 3 additional submitted papers, one in JCO that's in revision that will be accepted soon, as I will explain. And here is my take-home message of my talk. We need papers, we need projects, we need funding, but what is always more important is to focus our research on curing patients. We do not have to think in ourselves, we have to think on the patients. They are the most important. And here is another take-home message or recommendation or suggestion, mainly for the young PIs, but also could be for the older PIs like me. You can be a boss, you can be a leader, but if you are a leader, your contribution will be greater. Here I want to thank all my teams, in green the persons that are really involved in MDS, but the other persons also very involved in our research, and I want to thank them because without them, the research is not possible. As I explained in the beginning, my team is small, but we work very hard. I am happy with all of them. I'm very proud. And also I want to recognize the work done by my It's without her, it was not possible to do nothing with of research. Lourdes, Isabel Granada, Javier Grau, but Blanca helps us a lot in all the clinical information, all the clinical sampling of samples of cases. It's a mandatory person in of our research. Blanca and Lourdes are the PIs of another working group, but we work very well together, and it's like to work in the same team. And here, I just want to thank the organizations that helps us or give their support with money or with support during the last year. Agaur, Carlos III, Sociedad Española de Hematología, Rotary, International Working Group, Grupo Español de Citogenética y Hematología, the Josep Carreras Foundation. Without them, the institute also is not the same. The Stiftung, Asociación Española contra el Cáncer, del Grupo Español de Myelodysplasticos, Celgene, Celgene is the only company at this moment that have treatment against MDS, and until now we receive a lot of support from them. I hope to receive more support. Perhaps you know, but Celgene now was bought by Bristol Myers, so it's Bristol Myers Celgene. Perhaps the name Celgene will disappear soon, I don't know, but we think we have a very good relation with them, Celgene or Bristol, and IGTP also we have to think that until now they give their support. Thank you in all the different languages. I suppose that in Japanese is correct. If not, I will not recognize. And it's time for questions.
Okay, thank you so much, Kiko, for this nice presentation, for these very interesting conclusions at the end of the talk. So there is several questions related to your presentation, dealing with different aspects of the presentation. First of all, there is some interest in talking about the epigenetics, as you can imagine. So they're asking why DNA methylating agents are working in MDS. Do you know about the molecular basis of for the activity of azacitidine and other agents in MDS?
I think it's a question that do not have an easy answer because we know that ASA is a demethylating agent. But what is interesting to know that MDS patients, MDS disease is not a very frequent methylated disease. So still we don't know what this was. The idea is it works, but still we don't know the mechanism of how it works. Some previous findings suggest that it works more for toxicity than related with the methylation status, but it works. But you have to know that ASA works, but it's not a curative treatment. For a period of time, you treat the patient. For a period of time, the patient responds, but after some period of time, the patient relapses. And when relapse, you have no possibility of curation. It's very difficult to cure a patient. For this reason, I say MDS is a very beautiful disease. It's a very heterogeneous disease. And at this moment, it's difficult to have curation of these patients. For the reason that sometimes people say, no, I want to treat patients with, I want to do research in infant leukemia. In infant leukemia, 90% of patients are cured. And 90% of patients with MDS will die. We need to do more research in this disease and other diseases.
Okay. So beside the azacitidine, we have the opportunity to treat these 5q minus patients with lenalidomide. So you presented your data showing that the alteration in the expression of CK1 alpha could be related with the sensitivity of the patient to lenalidomide. So you know what, in lymphoma, at least, or in multiple myeloma, the mechanism of action of lenalidomide is very well described. It mainly depends on the cerebron E3 ubiquitin ligase. And as you know, CK1 alpha is a target of cerebron. So can we imagine that the cerebron activity and CK1 alpha are really relevant for the activity of lenalidomide in these patients? And is there any functional studies supporting this role?
If I do not have knowledge about that, I don't know if there are studies about that. It's a good point. That is something that for this reason, I wanted to show to all of you what we are doing, because we are very good in genomics, but we have not experience in functional studies. And I would like if other teams in the institution help us to complement our research in genomics with functional studies. For the reason I am open to collaborations with all of you.
Okay, so there is another question related to that point, to that this necessity of developing functional studies. So at least in MDS, there are only a few preclinical models which have been described so far. We know that there is no very truthful cell lines for studies. And do you know about PDX model, organoid model that can be useful, or some groups that have such models to collaborate with?
The problem of cell lines in MDS that these are just few cell lines that have MDS, but really are not from MDS patients, are from patients that have progressed to AML. This is a cell line when the patient have an AML. So to have MDS cell lines, I think there is one that started to be very useful, but really is difficult to grow in vitro in the culture. Another possibility that I am thinking also is to use mouse models to correlate MDS with the clonal hematopoiesis. And I know from a team in Madrid, is Dr. Javier Fuster, Josep Fuster, that have mouse models of patients with TET2 mutations, with NMA38 mutations. They study this for the relation with clonal hematopoiesis with cardiopathies, and we want to use these mouse models in the future or present to translate to the MDS study or MDS transformation of clonal hematopoiesis. Yes, it's a disease where the in vitro, the functional studies could be not so easy as other diseases. But I am sure that here in the institution are groups very strong in functional studies that could help me a lot in that.
Okay, so there is some questions regarding the chip. Why some people develop chip, and from data you have generated, why some transform to MDS? Do we know?
This is the question of a lot of millions of dollars. I don't know. it's a question that sometimes, every meeting, every week there is a meeting about clonal hematopoiesis, and this is a question that most of the speakers receive. At this moment, we don't know. But the best will be in the future, will be to try to know how to stop the evolution of clonal hematopoiesis, how to stop. What we know, if a patient has clonal hematopoiesis and mutations of TP53 and PPM1D, if you treat the patient with chemotherapy, this treatment makes that the clones with the mutation of the gene grows better and the normal clone grows worse. So with the treatment, you select the clone with the genes mutated. So perhaps if you treat a patient, what you are doing is selecting a population that is more aggressive. So it could be important to know if the patient have this mutated gene, perhaps not treat the patients with these treatments. With another ones, we need to discover, yes, but not with the traditional techniques, the treatments. It's something very interesting.
So regarding this,
sorry, if we live more than 150 years, all of us will have chip. And we plan to live a lot of years. I don't know, me no, but others yes. The younger yes.
We try to reach that. So regarding this, the influence of the chemotherapy in chip patients, so do we know whether the condition of chip increase DNA damage following the treatment, and or does it promote a selective pressure that increase the clonality?
What I read until now is the treatment selects the population with the clone with the mutated gene. So the treatment confers a selection of the clone with the mutated genes. This is what I know until now, that every week I try to read new papers and it's all this. But I am sure that in the next 10 years, we'll have a large experience. Perhaps you know that in Boston, by David Stimsak, that came here last year in IGC, in Boston, they created a unit of clonal hematopoiesis. which are involved cardiologists, hematologists, oncologists, genetic counseling. And this will be very, I think that we have to do the same soon in Spain. And inside the Spanish working group, they are trying to write a new guidelines for this group of patients.
Okay, so in the same context, is there any risk factor that we can identify in this study that you described of ovarian cancer patients? Any risk factor which can provide this development of secondary myeloid neoplasms following the chemotherapy or radiotherapy? So do we know first if it has to see with the treatment or if there is some risk factors?
What I have read until now is treatment per se is a risk factor, but for example, cases with ovarian cancer treated with platinum, these have a highest risk to develop secondary leukemias. And in those cases, they have one gene that is PPM1D mutated. That this gene is just mutated or normally mutated in secondary leukemias and very rare in primary MDS patients. So it's a gene very strongly related with the secondary leukemias and the previous treatment of this kind of treatment.
Okay. And regarding the genetic basis of MDS, do we know about something about the inheritance of MDS, about some genes that can be inherited?
Yes, this is something really new in the last 5 or 10 years that appear new genes that are germinal mutated, and when you have these genes germinal in all your cells, you have a high risk to have an MDS. This MDS is different than the typical MDS of adults because appear more frequently in children. MDS is very rare in children, but if you have germinal mutated genes, then you develop easily or more frequently an MDS of children. That is very, very rare. And now DDX41, some genes like that are known to be mutated in germinal patients or individuals.
And maybe a last question regarding the project funded by the International Working Group for the Prognosis of MDS, the ongoing project. So you are going to analyze the samples from many, many patients. So which kind of samples are you considering? Bone marrow aspirates or peripheral samples?
The best, if you want to perform if you want to analyze exome sequencing, is always to have bone marrow sample and peripheral blood from the same patient. This is the best. If you want to perform targeted sequencing with bone marrow or peripheral blood, also could be used. And in these 40,000 cases around the world, some groups send samples of bone marrow and peripheral blood. Some groups just send samples of bone marrow or peripheral blood. And this is a huge amount of information that soon, I hope that this ASH meeting will have the preliminary results. I hope that I will share with all of you as soon as I can.
Okay, so thank you once again, Kiko, for this very nice presentation, for all of this exciting new information about MDS pathogenesis and possibility of treatment. So thank you for your time.
Thank you. Thank you.
Goodbye.
Bye-bye.